We all start as a single cell, and yet we end up being an organism with 10 trillion cells with a lot of structure. And then during development, those cells are then sculpted in order to generate a specific structure that's important for function.
We studied this process of tissue folding in the fruit fly embryo, which is an excellent model system to look at the way in which cells can coordinate this collective movement. And so specifically what we were interested in is how mechanically can cells inside a tissue change the pattern of forces in the tissue to respond to different types of damage that the tissue might experience over the course of development. One way for cells to coordinate their behavior is through direct linkages with each other.
Actin and myosin are two proteins that are involved in a cell generating a pulling force. And so as a tissue is getting sculpted, the cells that compose that tissue are often pulling against each other in these chains in order to basically pull the tissue into the correct shape. So we had observed that cells were transmitting force between each other, but what we didn't know is how all of the cells in the tissue were interacting with each other as a whole in order to change the shape of the tissue.
And this was because it had been very difficult to quantify these networks at the scale of a tissue and then therefore understand their potential function during development.
And so what we needed to do that was to map how cells were pulling on each other globally across the tissue. And so this is where our collaboration with Jorn Dunkel's group in the math department was a breakthrough for us, because his group had been using an algorithm which had been used in astrophysics to map filamentous connections in galaxies.
And so what this algorithm can do is that, in this case, instead of tracing the structure between different parts of a galaxy, what it actually does is trace this network of connections across hundreds of cells in the tissue.
We discovered that there's a lot of redundancy built into the system.
Certain groups of cells can be damaged or not function as well, and the overall tissue still has a path that can connect the cells which need to go on, propagate force, and fold, and the overall folding can still occur correctly.
And so cutting individual linkages doesn't have a large consequence because the large-scale function of the net remains intact.
So this is exciting because this type of network of connections isn't limited to fruit fly embryos, but we see it across many different types of developing tissues.
And so by learning the mechanisms by which fruit fly embryos overcome obstacles, we're also gaining insight into possible sources for birth defects in vertebrate embryos.